In this video, we will be discussing our science experiment on creating an effective, non-toxic, eco-friendly weed killer. First, we will begin with the abstract of the experiment, followed by the introduction. Then we will introduce the hypothesis, the methodology of the experiment, the results with pictures and graphs, analysis, and whether the hypothesis was confirmed or disproven. And finally, we will discuss the conclusion. And now, I will start with the abstract. The importance of utilizing effective weed killer ingredients that are natural and non-toxic while aiding in the growth and protection of the surrounding biodiversity, while also providing an effective and eco-friendly solution that is safe for humans, animals, and beneficial to insects, soil health, and water ecosystems, is a vital consideration that can have long-lasting impacts on our agriculture, landscaping, and public health. The present experiment questioned, researched, and investigated the role of non-toxic ingredients to concoct a weed killer that is safe and does not harm the surrounding biodiversity. The concocted weed killer for testing phase 1 confirmed the hypothesis. Therefore, for testing phase 2, the weed killer materials and experimental procedures remain the same. Overall, the results for testing phase 1 and testing phase 2 confirmed the hypothesis that all non-toxic ingredients utilized for the concoction of a weed killer that is environmentally friendly and safe can be utilized as an effective weed killer that will not only inhibit the growth of weeds but will also kill the weeds and its respective root systems, while also providing an effective eco-friendly solution that is safe for insects, soil health, and the surrounding biodiversity. Weed killers, also known as herbicides, have had a significant impact on agriculture, landscaping, and public health. In fact, they have greatly benefited humanity in that herbicides have increased agricultural productivity, reduced labor intensity, have helped in soil conservation, and have improved aesthetics and safety of our environments. In addition, the added economic benefits of having an efficient herbicide that can reduce costs for farmers and landscapers, which led to more affordable food and services for consumers, has also had a positive impact. However, it is worth noting that such benefits come with a cost. The use of weed killers has raised valid concerns regarding their environmental impacts, their human health risks, and herbicide resistance in weeds. Many weed killers contain a chemical known as glyphosate. Glyphosate is known to affect the endocrine system, especially in the liver and kidneys, and is also considered carcinogenic, meaning cancer-causing. Popular and widely used herbicides such as Roundup and Cleanup combine glyphosate with other chemicals, therefore making it far more harmful to humans, animals, and the environment. In addition, these chemical formulas have not been tested for safety. In early April 2019, Monsanto, the company behind Roundup, was ordered to pay $2 billion in punitive damages and $55 million in compensatory damages to a 70-year-old man who claimed that his excessive use of Roundup caused him cancer. Many other people have also made similar claims that they have been diagnosed with cancer because of the Roundup weed killer. As of recently, it is estimated that over 750 weed-killing products use glyphosate. Due to this widespread use, glyphosate is in food, water, and dust, so essentially almost everyone has already had some type of exposure to it, though apparently not enough in significant numbers to reduce or stop the overall use of this chemical. Otherwise, cancer rates would reveal a significant increase, which has not been observed. Nevertheless, we see that the products that contain glyphosate, like Roundup, have also contributed to the loss of biodiversity as it destroys ecosystems. Glyphosate results in the reduction of different plant species in ecosystems, as well as in the reduction of various insects. This reduction results in the soil toxicity levels to increase. In addition to the prevalence of glyphosate and weed killers, other chemicals that may also negatively impact the ecosystem that are included in weed killers include atrazine, a chemical known to contaminate water supplies and that can be disruptive to aquatic life by affecting hormone systems, T4D, which can harm beneficial insects, aquatic organisms, and pets, and dicamba, which can damage nearby crops and plants. As a result, it is necessary to concoct a sustainable, effective, and environmentally friendly weed killer that utilizes non-toxic ingredients that aid in the growth and protection of the surrounding biodiversity, while also providing an effective and eco-friendly solution that is safe for humans, animals, and beneficial to insects, soil health, and water systems. As such, if vinegar is used carefully to target the weed, it will dehydrate and destroy a weed cell walls, causing them to wither and die while safeguarding soil microbes. Epsom salt dehydrates weeds by drawing moisture from their cells, while also minimizing harm to beneficial organisms and promoting a healthier ecosystem. Plant-based liquid soap breaks down the waxy coating on the weed leaves, which cause them to dehydrate while also reducing the risk of soil and water contamination, which preserves beneficial insects and microbes. Orange oil also breaks down the waxy cuticle of weeds that causes them to dry out and die while posing minimal risk to non-target species. The lemon juice lowers the pH on weed surfaces and burns them and inhibits their growth while also being eco-friendly. Finally, the mulch acts as a physical barrier that blocks the sunlight and prevents weed seeds from germinating while it enriches the soil and reduces the need for chemical-laden weed killers.
The goal for this experiment is to provide an effective and eco-friendly solution that does not affect the surrounding environment. This weed killer should kill the weeds within a span of 24 to 48 hours, and there should not be any remains after behind of the weed. However, if the first connected weed killer is ineffective, a new and improved weed killer will be planned out and connected for the testing phase 2.
Our hypothesis is as follows. If environmentally friendly and safe ingredients such as a combination of vinegar, Epsom salt, biodegradable and plant-based dish soap, orange oil, lemon juice, and mulch is used as a weed killer to inhibit the growth of weeds, then it will provide an effective and eco-friendly solution that is safe for insects, soil health, and the surrounding biodiversity.
The following materials were used: Lucy's distilled white vinegar, ½ gallon, Dr. Tears per Epsom salt, 1 cup, 7th generation dish liquid soap, biodegradation and plant-based, ½ tablespoon, handcrafted Blind's orange essential oil, 2 tablespoons, lemon juice from Meyer Lemon, ¼ cup, Munch, 1 cup, measuring cup, measuring spoons, 1 gallon multi-purpose sprayer. Step 1. In a 1 gallon multi-purpose sprayer, combine a ½ gallon of Lucy's distilled white vinegar, 1 cup of Dr. Tears per Epsom salt, ½ tablespoon of 7th generation dish liquid soap, 2 tablespoons of handcrafted Blind's orange essential oil, and a ¼ cup of lemon juice from a Meyer Lemon. Stir well until all the ingredients are completely dissolved and mixed. Step 2. Spray all the weed killers directly onto the weed, ensuring the weed is fully saturated. Then spread the mulch directly onto the weed. This serves as a herbicide that prevents seed germination. Step 3. Leave overnight and observe for the next several days. The independent variables in this experiment are the concentration of the weed killer, the application method for this weed killer, the time that the weed killer is applied, the environmental conditions such as temperature during the application of the weed killer. The dependent variables in my experiment are the rate of the weed's growth, the time it takes to see results, the weed mortality rate, the impact on surrounding plants, and the soil concentration. My controlled variables are the weed type and the surrounding cement. Our experimental observations revealed that testing phase 1 began on March 3, 2025, and presented a weed that was green in color with healthy roots. After spraying the weed with our connected weed killer, half the weed was covered in mulch to compare the difference between the half with the mulch versus the other half without mulch. A second observation of testing phase 1 took place on March 5th, 2025. It rained this day. The half part of the weed where mulch was added was observed to be yellowish-brown in color with desiccated roots. However, The other half part of the weed, without the mulch, appeared to be more yellowish than brown in color. As a result of previous success with testing phase 1, the same weed killer was connected for testing phase 2 on March 10, 2025 and utilized on the other weed. The chosen weed was observed to be green in color with healthy roots. After spraying the weed with our connected weed killer, mulch was added to the entire weed. The second observation of testing phase 2 took place on March 11, 2025. It rained this day. The weed was observed to be yellowish-brown in color and appears to be decaying with desiccated roots. Another observation took place on March 12, 2025. It rained this day. The weed was observed to be brown in color with desiccated roots. The mulch was observed to have spread around the weed, therefore likely preventing seed germination. The final observation took place on March 14, 2025. It rained this day. The weed was observed to be brown in color and the roots were observed to be desiccated and dissolved like a powdery consistence.
Overall, at the conclusion of testing phase 1 and testing phase 2, it was observed that both weeds changed from a green to a brown color, while testing phase 2 revealed a root integrity with a texture similar to sawdust on the 4th day of observation. It is evident that this is due to the multiple weed-killing ingredients breaking down the weed cell walls, causing the weed's root systems to decay to a point where it begins to turn powdery and dusty. However, it is important to note that for testing phase 1 and testing phase 2, it rained during the second observation date for both testing phases. This may have contributed to the dilution and potency of the weed killer, but it is still important to note that both weeds and their respective root systems successfully died, thereby confirming the hypothesis that a combination of vinegar, Epsom salt, biodegradable, and plant-based dish soap, orange oil, lemon juice, and mulch is used as a weed killer and as a means to inhibit the growth of weeds was an effective and eco-friendly solution, safe for insects, soil health, and the surrounding biodiversity. Finally, our conclusion. The findings of this experiment confirms the importance of concocting a sustainable, effective, and environmentally friendly weed killer that utilizes non-toxic ingredients that aid in the growth and protection of the surrounding biodiversity, while also providing an effective and eco-friendly solution that is safe for humans, animals, and beneficial to insects, soil health, and water ecosystems. Following the concoction of the weed killer, observations confirmed that weed death was taking place as a result of changes in color from green to yellow to brown, and the appearance of the weed's roots from healthy to desiccated. This was confirmed in testing phase 1 and testing phase 2. The hypothesis that if environmentally friendly and safe ingredients such as a combination of vinegar, Epsom salt, biodegradable, and plant-based dish soap, orange oil, lemon juice, and mulch is used as a weed killer and to inhibit the growth of weeds, and it will provide an effective and eco-friendly solution that is safe for insects, soil health, and the surrounding biodiversity was confirmed. Since testing phase 1 confirmed the overall hypothesis, the same ingredients and an experimental procedure were conducted for testing phase 2. Testing phase 2 also confirmed the overall hypothesis. Some factors in this experiment may have contributed to slight changes in the appearance of weed and its respective roots. For instance, the day following both testing phase 1 and testing phase 2, it rained. The dilution of the weed killer ingredients because of rainwater likely contributed to making the weed killer less potent. However, it's important to note that even with this unexpected factor, the weeds and their respective root systems still died. Future suggestions and modifications to the experiment would be accounting for outside influential factors such as rain by providing shelter and protection to the weed in a more controlled environment like in a greenhouse. This would allow for the weed to have a stable environment and temperature for both testing phase 1 and testing phase 2. For testing phase 2, twice as many observations were conducted. If the same had been done for testing phase 1, it would be easier to compare the changes in weed killer and the root consistency on a day-by-day basis against testing phase 2. Finally, maintaining a stricter observation schedule regarding the time of day these observations take place would have contributed to more consistency in comparing weed and root appearance against testing phase 1 and testing phase 2.